Is waiting for long.
Okay. So, I hope I'm audible.
Yes, you are.
Yeah, like unfortunately, I can't turn on my camera right now. I'm having some issues. Like I had to switch from the PC to my mobile phone. So anyway, let's start the session officially. So, hello guys. I'm Navneet. I'm currently a BTech Mechanical Engineering student at NIT. Just completed my first year and currently I'm in my break period. I'm also a student coordinator of UL Space Club. So, without any ado, let me directly welcome every one of you in the general. And today we are having a really interesting session. As we know, we started this journey long back in the COVID times where we used to have almost a session per every Saturday, every month. And later on, when we actually got back to the usual lifestyle, usual working style, we actually had to cut down the number of sessions, but it's actually quite wonderful. And it's worth noting that despite all the setbacks and all sort of difficulties, we are having a number of sessions going on even now, which includes actually covers a lot of topics ranging from say biology all the way to space science. And today we are having an eminent personality, Dr. Vinod Scariya, who has quite a lot of experience in the field of genomics and who has also been a part of Human Genome Project. So, sir, we welcome you to this session. I'd also like to welcome A.K. Kuti sir, founder and mentor of UL Space Club, Shajil sir, mentor, and all the dear participants who are here to listen to the speaker. So, without any further ado, let me hand over the spotlight to Shajil sir. Thank you.
Thank you, Navneet. We have a proud moment here. It's the 70th webinar of UL Space Club. The Space Club established and designed by E.K. Kuti, former director ISRO, and is under the umbrella of Puralangal Labour Contract Society, ULCCS, and it is running there for the past from the year of 2016. And we started this program during the COVID time when the people are kept apart, and that still we are continuing and connecting the scientists of India to our students. And most of the students really engage with the space activities. Now they understand that everything is connected and there is only one science, and we are integrating all the science areas to one and with a comprehensive view. And today we get a very, very relevant and very interesting topic with us. That's the genome and the personal genomes for precision medicines. And we are proud to have a great Indian scientist here, and especially he's from Calicut, and he's a role model for our students. And we are proud to say that he's connected with the UL Research Center at Calicut, and the director of UL Research Center, Dr. Sandesh EPA, is already joined with us. And before I'm going to formally introduce Dr. Vinod Scariya, I would like to invite Dr. Sandesh EPA for his few welcoming words. Welcome, sir. Sandesh, sir?
സർ കേൾക്കുന്നുണ്ടല്ലോ അല്ലേ?
Okay, sir.
ഗുഡ് ആഫ്റ്റർനൂൺ സർ. ഞാനങ്ങനെ ദീർഘമായിട്ട് സംസാരിക്കുന്നില്ല. ഞാനൊരു ജിനോമിക്സിന്റെ തുടക്കത്തിലുള്ള ക്ലാസ് കേൾക്കണം എന്ന് കുറെ കാലമായിട്ട് ആഗ്രഹിക്കുന്നുണ്ട്. ആ ഒരു അർത്ഥത്തിൽ പാർട്ടിസിപ്പന്റ് ആയിട്ട് മാത്രം പങ്കെടുക്കുന്നതാണ്. എന്തായാലും നമ്മള് ചെയർമാൻ കൂടി പങ്കെടുക്കാന്ന് പറഞ്ഞിരുന്നു, പക്ഷെ ലാസ്റ്റ് മിനിറ്റ് വേറൊരു മീറ്റിംഗ് കയറി വന്നു. എന്തായാലും നമുക്കിത് സ്പേസ് ക്ലബ് അടക്കം ഒരുപാട് ഇനീഷ്യേറ്റീവ് ULCCS ഉണ്ട്. സോ വി തിങ്ക് വി ക്യാൻ ഹാവ് മോർ സെഷൻസ് ലൈക്ക് ദിസ്. എനിക്ക് തോന്നുന്നു വിനോദ് സാർ IGAB-യിലുള്ളപ്പോ ഒരു ഫൗണ്ടേഷൻ കോഴ്സ് തന്നെ ചെയ്തിട്ടുണ്ട് ജിനോമിക്സിൽ. അപ്പൊ അതിന്റെ ചില ഡിസ്കഷൻസ് ഒക്കെ ഒരു ഘട്ടത്തിൽ നടന്നിരുന്നു. ആ അർത്ഥത്തിലുള്ള പരിപാടികൾ ഇനിയും നമുക്ക് ആലോചിക്കാവുന്നതാണ്. സോ താങ്ക് യു വിനോദ് സാർ ഫോർ joining this initiative.
Thank you, Dr. Sandesh EPA. And for our students, let me introduce our scientist, Dr. Vinod Scariya. And Dr. Vinod Scariya is a clinician turned computational biologist. His research interests are understanding the function, organization, and regulation of vertebrate genomes and how genomic variations could potentially impact them. He is the co-founder of Genomics for Understanding Rare Diseases, India Alliance Network, that's the Guardian. That's the organization which is one of the largest network of clinicians and researchers in India working on rare genetic diseases. His research has contributed to a number of insights and technologies which are widely being used in India for diagnosis and screening of genetic diseases. He has been part of collaborative genomics and projects aimed at understanding Indian diseases. He has been part of collaborative genomics projects aimed at understanding Indian and Asian genome diversity. He has also been part of the early whole genome sequencing population genomics projects, including Indian, Sri Lankan, and Malaysian genomes projects and population genomics projects, including and is a member of Hugo Pan Asian Population Genomics Initiative. That's Hugo Papigi Task Force. He has also established one of the earliest fellowship and education program in genomics for clinicians in India. The COVID-19 pandemic has been his lab delve into the genetic epidemiology of SARS-CoV-2 in India. His lab today hosts a major resource for enabling genetic epidemiology as well as functional interpretation of SARS-CoV-2 genomes and genetic variation. He is also spearheaded the state-level genomic surveillance program in India. His research has also contributed to the earliest reports and significant understanding on the immune escape variants in COVID-19 reinfections and vaccine breakthrough infections. He has also been a propounder of open data approaches and has been well quoted in national and international press for his work in this area. Dr. Vinod did his undergraduate from medical education from Calicut Medical College, University of Calicut, and PhD in computational biology from University of Pune. Vinod has over 200 research publications in international peer-reviewed journals and 2 books to his credits. He is also on the editorial board of a number of journals, including PLOS, PLOS One, Translation Genomics, Journal of Orthopedics, and Peer J, and International Journal of Rheumatic Diseases. He is the recipient of CSIR Young Scientist Award for Biological Science in 2012, a Cavill Frontiers of Science Fellow of the US National Academy of Sciences in 2015, and elected Fellow of Royal Society of Biology 2017 and Royal Society of Public Health in 2022. And his core scientific area is in genomics, genome informatics and computational biology, genomic surveillance and viral genomics. And he's an editorial board member of Frontiers System Biology, PLOS Ones and Journal of Orthopedics and International Journal of Rheumatic Diseases. And he's a recipient of several awards and Fellow of Royal Society of Public Health, Fellow of Royal Society of Biology, and gold medal in biochemistry in Medical College, and is a gold medal in physiology in Calicut Medical College, and GeneCon 2000 National Biochemistry Quiz. And really, he's a role model of students. Most of them are searching for the medical education, but his life and his life story shows that there is a large way and a great way is there for our medical students to engage with the research activities for our future and especially for our health, and that's connected with the health. And genomes are already established and defined, and we are thinking about our existence on this earth, and we are thinking how to design the drugs that's suitable for a particular person with a particular genome, or a genome hierarchy is there. So without taking much more time, I invite and proudly invite our chief guest, Dr. Vinod Scariya, to this forum, along with all the veteran scientists, students and teachers gathered here to hear this interesting lecture. Welcome, sir.
Thank you very much. It's a pleasure and honor to be here. I understand that this is largely a space club, and probably the first time you'll be listening to something in biology or something in genomics. Thank you very much for Shakil sir for inviting me here, and thank you very much for Dr. Kuti for organizing this fantastic session. I've heard a lot about it, and it's a privilege to be here. I hope I'm audible and my slides are visible.
Yeah, slide has come.
Right. So over the next 30 odd minutes, I will briefly describe about genomics, or personal genomes, and how it is going to change the way we practice medicine. And the first time Shajil sir asked me to talk about genomics, I readily agreed to it because as school students who I understand are the majority of the audience in here would be probably the first generation of people who would use routine genomic sequencing in their lifetimes. Now, all that I'm going to talk is work done by a fantastic set of people, mostly from my previous institute, the CSR Institute of Genomics and Integrated Biology, and a fantastic set of colleagues and collaborators spanning across the country and across the world, and consistent funding across the world. Now, before I start to talk about genomics and how it's going to change the way we practice medicine, I will give a very old story. And the story was from 1971. And in the 1970s, when clinicians used to practice medicine, and if they wanted to look inside the human body, for example, an individual came with a pain in the stomach or the pain in the head, the only way to really look at what really is wrong with the patient is to actually open up the human body. And so that would mean that surgeons would perform a surgery to open up the gut or open up the head to identify why is the pain out there. Now that would necessarily mean that a significant large number of people would die out of this procedure. Now, quickly in 1971, this engineer, Godfrey Hounsfield, who was largely in the music industry before, has nothing to do with medicine, had a bright idea that you can actually use X-rays, and in a particular fashion, if you use it, you can pretty much reconstruct what is inside the human body. And he created this very interesting contraption in 1971, what we today call as a CT scanner. And with a CT scanner, the way to practice medicine has changed quite significantly over the last half a century. Now, when I was in medical college, CT scans were just arriving in the medical scenario, but 25 years later, you would realize that CT scans have become a commonplace. In fact, every Malayali out there would know what a CT scanner is, and if you require, where to get a CT scan from. And of course, the prices of CT scans have also dwindled very rapidly. To that extent, that CT scanners today are, in fact, one of the main investigations that are being used in medical practice. Now, why did I really tell about CT scanners? I told about CT scanners largely because of the fact that CT scanners allowed clinicians to peek inside the human body for the first time without having to open up a human body. So without having to do a surgery, you can really know what really happens inside the brain. Was there a block or whether it's a bleed? And you can actually figure this out without even opening up the skull of the human body. Now, why I'm talking about this is because today we have the ability to peek not just inside the human body, but also inside every one of the 37 trillion cells in terms of what is the software that we have. I'm talking about DNA. In fact, every one of us have cells. A human body is approximately around 37 trillion cells. And all of these 37 trillion cells have pretty much the same software. And the software is what we call as the DNA. And the DNA is organized into 23 pairs of chromosomes. 22 of them are autosomes, along with X and Y. And in total, this encompasses around 3.3 billion alphabets of A, T, G, and C packed inside the human cell. And each of these 37 trillion cells originated from one cell, and therefore they have almost exact copy of this 3.3 billion base pairs. Now, in total, this 3.3 billion base pairs is what we call as the human genome. Now, of course, this was an enigma until very recently, but of course, researchers has allowed us to understand what does this software do. So it is essentially like a book, a recipe book, And recipe books would have recipes for each food items. Each page would be, for example, recipe for example, chicken biryani, or to make a mutton fry, so on and so forth. Or for that matter, you can even make a cake. Now, each of these pages is what we call as a gene, which has an instruction to make a recipe, and the recipe outcome is what we call as a protein. So essentially, you have the genome, which is a recipe book, 3.3 billion alphabets in the genome. It is encompassed into approximately around 20,000 odd recipes, and each of these recipes make a particular item, and this item is in the genomic parlance what is a protein. So essentially what a genome does is it encodes for around 20,000 proteins, but of course that's only one component of it. Today we know there are probably 100,000s of genes which encode for non-protein coding genes. I'll come to that probably in a later talk from now. But for all practical purposes, we have a genome of 3.3 billion alphabets which encode for approximately 20,000 gene encode for proteins. And every one of our 37 trillion cells have pretty much the same copy of this material, and therefore has the instructions to make the recipes that each of the cell requires to make. Now, in 2003, we are fortunate that for the first time, we are able to read this alphabets, all 3.3 billion alphabets, not all 3.3, but approximately 2.8 billion alphabets in what we call as the draft human genome. And this was a very large program called the Human Genome Program, where a number of countries participated, approximately 20 odd countries, they participate with research institutes and decoded the almost 3 billion or 2.8 billion alphabets that found the human genome. And this was indeed a landmark because for the first time in the history of humankind, it could now read the blueprint of what a human is. Now, what really happened after that, 2003, was even more remarkable, that the cost of sequencing a whole human genome started dwindling extremely rapidly. Now, to put into perspective, we have on the x-axis is the year, and what you have in the y-axis is in the log order of cost per million basis of sequencing the genome. Now, it is estimated that the Human Genome Program costed approximately $3.3 billion. That is approximately $1 per nucleotide. And today we are looking forward, and it's already in this site that you can sequence a whole human genome for less than $100. So $3.3 billion for a human genome. Today we can sequence a human genome for less than $100. And of course, that would necessarily mean that in the next 1 decade, genomic sequencing will become affordable and accessible to a lot more people. And you as students would be the first generation of individuals in this world who would benefit from this remarkable new technology. Now, what really happened with this reduction in the cost is that the ability that human genomes, and for that matter, many more genomes of organisms around us, could be sequenced at scales which were never thought about before. To put things into perspective, the countries in this world who are planning or have planned or are sequencing entire individuals in the country. There are countries which are sequencing millions of whole human genomes of individuals in that country. And that would necessarily mean that in a decade's time, probably we will reach 500 million whole human genomes available to mankind. And that, in my opinion, is going to really change the way clinicians practice medicine, the way we understand humans and human diseases. I'll take you back to some time back in 2008. That is when I started my lab. There were not many human genomes which were available at that point in time. To be exact, there were only 6 human genomes available in 2008. And the chart on the top is essentially the diversity plot or the admixture plot of the world populations. Now, each of the small lines is actually an individual in that particular population. Each of the block is a population by name. And the color necessarily represents how are they admixed or how are they intermingled with each other. Now, as you see here, a significant large proportion of the world population is less admixed or quite homogeneous in many aspects. Like, for example, if you look at East Asia, if you look at Africa, if you look at Europe, there's much lesser admixture. But if you look at the middle portion of the chart, which is the Middle East and Central and Southeast Asia, you would see a very significant admixture of human population. That means that populations of different kinds sort of married together and produced offspring with the diversity of the genomic material. Now, unfortunately, if you see, almost all human genomes at that point in time came from very homogeneous or less admixed populations. Whether it is the reference genome, which frequents or centered the Watson genome, the Japanese, Chinese, or the Korean genomes. And a significant portion of the world, which approximately half of the world population, which is highly admixed, had absolutely no representation of the human genome. So that's when we started thinking, can we not do it in India? So we announced the first Indian genome in 2009, December 8th. This is part of an announcement in the Indian Parliament by the then Honorable Minister of State for Independent Charge for Science and Technology, Shri Prithviraj Chavan. And we didn't really stop with one. We went around these countries around us to also sequence initial genomes from these countries, including the post Sri Lankan genome. We are part of the first Malaysian genome, the first Philippines genomes. And we're also part of the Human Genome Organization and Asian Population Genomics Initiative. And today in our lab, we have one of the largest data sets of human genomic diversity, encompassing close to around 4,000 odd genomes and exomes, encompassing Southeast Asia, Middle East, and much very recently with the indigenous program, 1,000 odd whole genomes from the Indian diverse populations. And the question is whether it's not whether you can do genomes or whether you can analyze genomes. How can we really change the way we practice medicine? Now, that is a big problem in India because there is no one genome that we can call India as representative. Because India is a very large country. Today, we encompass almost a sixth of the global population. And more importantly, we are a very, very diverse population. Put numbers, we have approximately around 4,600 odd communities. They're largely small or large endogamous groups. We have around 325 functional languages and 25 recorded scripts. And therefore, we are a quite very well diverse country. And therefore, it's unlikely that we would find a genome that is representative of the country like many other countries or populations. That is a new challenge because not just a challenge because we're large and diverse, but also a challenge in our ability to understand genomics and put it back into clinical perspective. So to put things into perspective, we have a very large country with a population of approximately 1.3 billion to 1.4 billion, a sixth of the global population. But apart from all the diversity that we have, the most important thing is, unlike many other countries across the world, we have largely been undisplaced by wars or famines in the last 2 to 3 centuries. That would necessarily mean that everybody in this country will be able to approximately trace 3 to 4 generations of their ancestors without much of an issue, because we all know where our grandparents lived, who our relatives are, because they have largely been undisplaced by war or famine, which most of the rest of the world has gone through in the last 3 odd centuries. And a large country would necessarily mean that every rare condition that you could ever think about, when we think about rare conditions, we say 1 in 1,000. And we would have 1 million people when we talk about a prevalence of 1 in 1,000. I'm not sure you would appreciate this fact that an ultra-rare genetic disease of 1 in 1 million prevalence, we would have 1,000 odd patients with that disease. So that would rather mean that every possible disease of genetic origin anywhere in the world, we would have them. And most importantly, we would have them not 1 or 2 or a dozen, we would have them in thousands in numbers. And that indeed is a fantastic, great opportunity. And when we talk about these opportunities, we should also talk about extremely rare genetic diseases, because many of these are extremely rare in origin. Even 1 in a million, I said we would have 1,000 odd patients with a genetic disease. And many of these patients do go through multiple rounds of physician visits, multiple rounds of misdiagnosis, and a considerable amount of time, energy, and money is lost before even they can get into diagnosis. And in fact, a very small percentage of individuals even get to a molecular diagnosis. And to put these numbers together, there are around 70 million people in India who would have a genetic condition, and many of them do not understand that they even have a genetic condition. Now, 70 million is approximately twice the population of Kerala, if you put it in perspective. And that would necessarily mean that even within this webinar, there are individuals who have an undiagnosed rare genetic condition. Like, you might have a rare genetic condition, the clinicians of the family might not have diagnosed it, but of course, by virtue of the fact that you have a genetic mutation, you do have the genetic disease. So how do we really uncover this disease? So what we did in 2015 with my colleague Dr. Sridhar was to essentially go to clinicians, very normal, routine clinicians who work in clinics, who work in hospitals, and sort of educate them and tell them, if you have a familial genetic mutation, that means there are 2 or more siblings or 2 or more blood relatives in 3 generations who have the same clinical feature, could you refer them to us? And today we work with approximately 300 odd clinicians from around 70 clinical centers across the country, and of course outside the country in some nearby countries, to refer patients who have a familial undiagnosed genetic condition. And what we do in the lab is to essentially take them up as part of this program called Guardian, which is funded by CSIR, to take these patients, do a genomic analysis, and get back to the clinician and tell that we have found the molecular defect, what caused the disease, and you can treat the patient accordingly. And of course, to do this, we also need to step into the lab and do a variety of experimental studies to prove that that variation indeed, of the mutation indeed caused the disease. And therefore, we can go back on a routine basis back to clinician in approximately 50% of our cases to be able to tell the clinician that I think we have found the cause of this patient, the molecular cause of this patient, and we can treat the patient accordingly. And this is not in ones or dozens. We've done this in thousands now. And in 2019, we published this paper with approximately 280 clinical colleagues and authors, sort of summarizing of experience working on a number of genetic diseases which were new to India, discovered new in India, and affecting millions of people together. So we have taken it from educating clinicians, doing research, to diagnosing patients, to now today a national rare disease policy. India indeed today has a national rare disease policy based on a large amount of evidence which is created as part of this program. I'll probably just give an example of how this program works and what is the real impact of this program. And in 2015, when we put together this program, this was an article which appeared in news. And this news essentially stated about a parent who asked the president of India permission to euthanize 6 out of their 8 children. And this is a photo that came in the news article. The family is from Agra, and the children have a progressive neurological disorder. Until approximately around 3 to 4 years of age, the children are perfectly normal. As you see here, there's a child who's perfectly normal and standing up. But as they grow in age, they eventually lose their milestones. They cannot stand up, and eventually they end up in a vegetative state and they die. And they don't have a diagnosis, they don't have a cure, they don't have hope. So this came to us because euthanasia is not permitted in India by law. So this came to us to avail a diagnosis. We did a genomic analysis of multiple members of this family of parents, and we found an extremely rare genetic mutation. confirming the diagnosis of cystic megalencephaly. Of course, the scientific term for this disease is quite long. It's megalencephalic leukoencephalopathy with subcortical cyst type 1, an extremely rare genetic condition. Now, of course, for every other study, this is the end of it because you have found the mutation, you have found the diagnosis. So let's really stop and move forward. That's not what we did. What we did is we went to this family in the community and we were asking around, and we found that this family was a quite consanguineous family belonging to a community called the Nalband. And just by talking to community leaders, we realized that there are probably many more patients for the same disorder in the community. So we did a house-to-house, door-to-door survey, scanning around 100 odd individuals. And what we found was enormously surprising to us, because approximately 28% of the individuals in this community had one of the 2 copies of this gene abnormal. And that would roughly translate that 1 in every 500 children or 600 children born to this community would have the genetic disease. Otherwise, an extremely rare genetic disease elsewhere in the world. And by doing a door-to-door survey, we indeed identified another 2 families with 3 children affected with this particular genetic disorder. So that necessarily means that this is not a rare genetic disorder anymore. For this population, that would be a prevalent genetic disorder because it affects 1 in approximately 500 to 600 live births. Therefore, you need to have a diagnosis which the patients can access. And more importantly, in a consanguineous community, you want prospective parents to test whether they carry 1 or 2 defective copies of their genetic event. And if they have 1 copy each, then we know that the offspring would have a 1 in 4 chance of developing the condition, and then they could be counseled appropriately. So right from the first case referral to the diagnosis, to a publication, and in fact, diagnostic analysis, which goes under the name MLC1, Montanap, and Nalband mutations. Both of them are founder mutations, which are identified in India for this particular genetic disorder. In 3 years, we can take it from the patient to the diagnostic, impacting a large number of people across the country who belong to this community called the Nalband. Now, of course, we were surprised why did this family or community will have this genetic disorder, because this genetic disorder was the only other case of this genetic disorder for this particular genetic mutation was described in the Middle East. And through an extensive survey of the community, through the history of the community, we realized that this community traces ancestry to the Middle East. And that necessarily means that there have been Indians for generations altogether, hundreds of years, probably 3 to 4 years. And they have been Indians in all aspects. But nevertheless, they have a small portion of the genome which traces their ancestry to a different part of the world. And this is not really something new. There are 10 million individuals in India who trace their ancestry to the Middle East, who over many centuries before has come in as traders, has come in as friends and travelers, and much later as invaders. But nevertheless, they all carry a small portion of the genome who trace their ancestry to a different part of the world. And therefore, they might have disorders which are very prevalent in a different part of the world. And therefore, clinicians in India might not be best equipped to understand those diseases. And therefore, we need to extend this fatherhood, not just in terms of our culture and practices, but also in terms of our ability to look at genetic diseases. Now, this is just an example of people who have traveled from the rest of the world and became Indians over many centuries. We also have many Indians who traveled to the rest of the world and was become members of each of those countries where they traveled to. And that is a much larger Indian diaspora. So that necessarily means that looking at human genomes in India would help a lot of people outside India, including people who traveled from the rest of the world and people who traveled from India to the rest of the world. And that would necessarily mean that genomics is something that should be based on brotherhood and collaboration, because what we identify in India could be beneficial elsewhere, and what could be identified elsewhere could be beneficial to Indians living in India. Now, I said about a rare genetic disorder which is very prevalent. Now, that opens up a very big question on what is really prevalent. When you talk about genetic diseases, people will always say that, okay, that is so rare, why should we really work on it? Now, how do we really know what is really prevalent? Now, there's a problem here. The problem here is that all prevalence estimates is based on the fact that clinicians diagnose diseases and report them. Now, what if clinicians don't even diagnose diseases, right? Like the case that I sort of explained, they never had a diagnosis, and therefore they would never be part of any statistics. Now, how do we really change this paradigm? You cannot really educate all clinicians to now say that let's all diagnose genetic diseases. Now, that's not really possible. The only other possibility is to be able to sequence individuals who are apparently normal, like you and me, who don't have a known genetic disease, but apparently normal and healthy. Then we sequence genomes of individuals, and all of us have 2 copies of their genomes, and there might be defects in just 1 copy, so therefore we don't manifest the disease. But by chance, people in our blood relation who could become double copy defective could now manifest the disease. Now that is really possible, and therefore you can look at population scale genomes to understand what is prevalent in the population. In fact, what disease is prevalent in the population. And if they're not prevalent by clinical estimates, that means that there's a huge gap of diagnosis of that particular disease. By educating clinicians on that gap would allow patients to be diagnosed very efficiently. So until 2019, we never had the possibility of doing this. So in 2019, CSI had funded us to do 1,000 odd genomes. And of course, 1,000 odd genomes in 1.4 billion people is really nothing. We need to probably do probably 1 million odd genomes in India to be able to get to the bottom of it. But nevertheless, the 1,000 odd genomes is extremely useful because it can identify extremely prevalent genetic conditions. And we needed to prove that you can indeed identify such diseases which are extremely prevalent. Now, how do we do that? You could after the genomes and ask this question, what is the prevalence of the genetic disorder? And what we realized is that there are many genetic disorders which were never thought to be very prevalent are indeed extremely prevalent in the population. And there is one example of it we recently published. This is a condition called familial hypercholesterolemia. And I'm sure everybody knows what cholesterol is. There is HPL, LDL, VLDL, and a variety of different cholesterols in the human blood. And we all test for cholesterol quite routinely. But what we don't do is beyond a cutoff of cholesterol, the likelihood that these are familial cases and the likelihood of clustering of high cholesterol in families. And in fact, people discount this because they might be eating the same food, they are the same family, they have the same lifestyle, and therefore they have the same high cholesterol. But in fact, a significant large number of individuals in our population, approximately 150 individuals, do have a condition called familial hypercholesterolemia. And in fact, today we know what are the genetic variants which are prevalent in the population. We have today now traced out families where there are multiple members who are affected with high cholesterol. Now, how is this important? It's important, one, because it's extremely prevalent. And second, it is important because this is a treatable genetic condition. An adult with high cholesterol is unlikely to be effectively treated, but a child with high cholesterol would be effectively treated with a very cheap medication. And these are statins. Now, what does it mean? It means that if you have a family with familial hypercholesterolemia, you could check for the cholesterol levels or the genetic variation in children who don't manifest the disease yet. They don't have a heart attack yet, but they will get a heart attack when they reach the 20s or 30s. But to do that can be identified early before the clinical, and can be now treat them very effectively. And to treat them, of course, we treat them using statins. And statins could also cause an adverse event called statin-associated myopathy. And approximately a third of Indians do have a genetic variation which could predispose them to statin-related adverse events. So you could also test for the genetic disease You could also test for the genetic variant, which can cause adverse event. You could also diagnose diseases which never were thought to be diagnosable or treatable, and there is one such condition called primary immunodeficiency disorder. There's a cohort that was built in Government Medical College in Calicut. The people who interaction with the pediatrics department was instrumental in building this. And this genetic disorder would mean that children have no patent immune system, and therefore they would have infections, and infections would end up with ICU admissions and eventually death because they don't have an immune system. But if you can diagnose them very early on, typically when they're just born, you could treat them very effectively by either instituting them immunoglobulins, IV. So you can administer probably every month a dose of immunoglobulins, or you can change your bone marrow, what we call as bone marrow transplant. And today there are dozens of children who have access to very accurate treatment and are living perfectly normal lives. And in fact, one of them has actually joined an engineering course and probably passing out this year or next year, despite having the fact that they would not have survived if the diagnosis was not done early on. So therefore, a genetic diagnosis can actually make a lot of difference to patients because they have a hope to live and they can be very productive members of the society. There are many other disorders which are now very prevalent in the population. This is something called sudden cardiac death or cardiac channelopathies. Approximately 1% of Indians do have the genetic mutation. And of course, you do see news articles of people who ran after a bus and collapsed, dying. People who went to gym, exercised and collapsed, dying. Young individuals who are apparently healthy, died suddenly in sleep. These are all because of cardiac channelopathies or sudden cardiac death, as we call them. And this is because of a genetic mutation, which is very prevalent in the population. And if you know that you have this genetic mutation, then you are supposed to be devoid of very strenuous activities which can precipitate cardiac arrhythmias and death because of cardiac arrhythmias. I'll stop there and explain why this is even more interesting now. It's even more interesting now because sequencing costs have been dwindling very rapidly, and you can do it much faster today. It took almost 2.5 decades for the human genome program to be initiated and completed. But today you can do a whole human genome sequence in less than 24 hours, and at a cost which is increasingly becoming cheaper. And that would necessarily mean that it would, in the future, probably a few years in the future, become one of the first lines of genetic testing for rare, undiagnosed genetic diseases. So we do have a paradigm shift then. That would mean that today what genetic testing is done as an explanation identifies a patient with a possible clinical phenotype or clinical disease, and you do a genetic testing to confirm the diagnosis. But tomorrow the possibility is that you can do a sequencing of individuals, probably newborns, and then be able to ascertain what is the genetic disease that they have, and depending upon the genetic disease, be able to treat them before they manifest the clinical symptoms. In other words, you can have a very proactive way of treating individuals who are at high risk. Of course, there are multiple examples of how we could do this. For many of the examples that I sort of described, by early detection of individuals at risk of developing the disease, you could institute very precise treatment to these individuals, and they can be perfectly normal individuals contributing to the society. The other aspect is, of course, a whole genome sequencing, which means that you don't have a variety of genetic tests anymore. Today, you have a variety of kinds of genetic tests for chromosomal abnormalities, for deletions, for duplications, for repeat expansions, for small insertion deletions, for single nucleotide variations. They're all different tests today. But the whole genome sequencing can encompass all of these kinds of variations and can actually do it much faster in less than 24 hours. So that necessarily means that eventually all this will go away and you will have just one genetic test, which is a whole genome sequencing. And more importantly, if you have a whole genome sequencing at birth, you can use the genome very differently during the course of your life, because your genetic material is going to be constant from the day you're born to the day. But depending upon the as you are in, you could now use this genomic sequencing to address very different kinds of disorders which appear in very different points in life. And therefore, genomic sequencing is for a lifetime. And with the advent of newer technologies, or largely for that matter, artificial intelligence, we are now able to look at diseases at much higher, much better efficiencies. And this is one of our recent publications called Wilson Gen AI, where we have used artificial intelligence to be able to sift through billions of variants to be able to identify variants which can cause genetic diseases. And more importantly, you need to communicate this information back to the clinician very quickly. You don't expect the clinician to be holding a hard drive of a few terabytes to be able to read your genomic data, but you need to have an efficient communication system which can communicate in a scalable, secure, privacy-protected methodology how the evidence that the clinician needs to be acted upon is communicated in an efficient, timely manner. And this is something that we launched in 2019. It's called the Genome Card, which is now co-developed as a product. It's called Pocket Gene. In the future, you will have a credit card type device or a card type card, which you can swipe or the clinician access the relevant information using information that can be accessed during the process of investigation. So you can do a whole genome sequence and the clinician can mine the necessary evidence at the point in time when he or she requires it the most. I'll stop there, but before that, let me conclude by saying genomic sequencing is becoming extremely affordable for routine clinical implementation. Within half a decade, that is going to be part of routine clinical implementation. Sequencing can enable disease diagnosis of individuals who don't have a diagnosis, and a population-scale genomic data can actually help understand what the prevalent diseases in the population so that we can prevent them, we can educate clinicians to treat them, and more importantly, we can eliminate them in the future. Many of the genetic diseases are very prevalent in the population. That would necessarily mean that approximately 1 in 10 individuals have an undiagnosed genetic condition. We can make it diagnosed by using genomic sequencing. And therefore, genomic sequencing is able not just for patients, but also for healthy individuals who can identify the risk of developing a genetic condition and be able to stop the disease in its tracks. And more importantly, a genomic sequence is for a lifetime because the genomic sequence is going to be constant from the day you're born to the day you're dead. So I hope that you are the new generation of individuals who would access this technology to its maximum. Thank you very much, and I can take questions.
Thank you, thank you. It is indeed a what do you call a blessing that we have been able to get Dr. Vinod Scariya to make a presentation on a topic which is highly futuristic and which is going to guide our future generations. That means the present student generation, which he started saying that they are going to be the beneficiaries of this new approach and new technology, and environments of his research and his past achievements, reassuring us that this technology, this approach is going to guide the human in the generations to come. In fact, I heard how it is beneficial, and particularly immunodeficiency, cardiac encephalopathies, and things of that variety. And a bright future I see lying ahead for this scenario of genomics. Genomic scenario is a bright future, and our students are actually blessed to have got an opportunity to listen from the horse's mouth about the developments that is taking place in this area. And there, if they wish to study more, understand more, they will be able to get, I hope, guidance from Dr. Vinod Scariya and Dr. Sajil. I congratulate him for identifying and bringing in the midst of us Dr. Vinod Scariya. And thank you very much, sir. It is indeed a rare opportunity for us to get a glimpse of what's happening in this area and what we can expect out of it. Thank you very much. It's a nice day and a nice webinar. Thank you very much. Is he still there or left?
Hello, good talk. Yeah, good talk.
Shajil sir, I think we can proceed with the students' question.
Yes, sir. Am I audible?
Yes, you are.
Yes.
Hi all, I'm Shirin Ziba, and I have completed my BTech in Electrical Electronics last day. So this is the question and answer section for the 17th webinar. How can we protect individuals from potential misuse of genetic information?
Yeah, so the only way you can prevent the misuse is by appropriate legislation that needs to be brought in, so that information is not misused and becomes detrimental for the individuals. And we do have similar legislation for a lot of things that happen around us. Like, for example, we have a legislation which covers Aadhaar and its misuse. We have a legislation which covers banking, that means the transactions in banking and its misuse. And very similarly, what we need urgently, very urgently, is that we need to have a similar legislation which will prevent misuse of genetic information. We don't have it at this point in time, but as more and more people become aware and individuals start doing it, what is really more important is to have a legislation which will prevent misuse of genetic information. Many countries do have implemented this, and many countries are today implementing it in the next few months. But I think with the new legislators that we have sort of elected, we should look forward to similar legislation in India also.
Thank you, sir. So next question is, how can machine learning algorithm be trained to interpret complex genomic data effectively?
Yeah, so sort of machine learning that I sort of described involves two things. One, it actually looks for genetic mutations which cause diseases, and it learns from what are the kinds of genetic mutations which can cause disease. And using that model, it can go back and look at billions of genomic variants and be able to say that, okay, this genetic variant looks like it will cause it. So that's number one. Number two is, apart from all this, there are new de novo ways of looking at how evolution of mutations do happen. Evolutions and mutations by definition occurs in a random fashion, but by selection, there are prevalence of certain kinds of mutations that are more frequently occurring. So machine learning can actually sort of give a glimpse of what is the likelihood of a particular mutation to be selected positively or negatively. And this can sort of allow us to look at a variety of associations with genetic diseases, not just variants with genetic diseases, but also variants that can sort of reduce the effect of a genetic disease.
Okay, sir, thank you. And next question is, how can precision medicine contribute to the goal of achieving personalized healthcare for every individual?
Yeah, so what I would imagine a dream is public Kerala is one of the first states where every newborn, at least in the government medical colleges who are born, or the government system which is born, undergo a number of tests which is called a newborn screening. I think there are 6 tests that are being administered at this moment in time. And a newborn screening test, or NBS, necessarily means that you can diagnose some children who can be treated very effectively, provided they can be diagnosed very early. Or in other words, when you test them at the newborn stage, you can diagnose them very early on before they manifest the disease, and you can treat them. Right? Now, there are only 6 tests that we currently offer. For example, the US has around 37 tests that they offer. But of course, still limited number of tests that they offer. A whole genome sequencing tomorrow can sort of get away with this problem of a limitation in the test, because once you have identified every possible genetic variant that causes a disease, and by sequencing an individual, you can figure out what are all the genetic diseases that can come up. So even if your test is not included in your 6 or 35, you can still identify a genetic disease which is treatable. And today, approximately around 400 genetic disorders which are treatable.
Okay, sir, thank you. And how do parents and families perceive the value and risk associated with newborn screening?
Yeah, I think that is where education is more important, because the perceived value is largely because of the fact that many of these disorders are treatable. There's no point in telling a parent that you have a disease which is untreatable, right? It doesn't really add any value to the test. But if you have a disease that is treatable and effectively treatable, like for example, when I talk about familial hypercholesterolemia, by identifying a newborn with familial hypercholesterolemia, and this year the EU has instituted a program for newborn screening for familial hypercholesterolemia. But studies have shown that you can save 20 years of the productive life of an individual. The individual would otherwise have a heart attack by in the 20s or 30s, but by instituting early diagnosis and early treatment, very effective treatment using statins, you can prolong the life, the productive life, not the total life, the productive life of an individual by around 20 years. And I think that is very, very remarkable. Right? Instead of having a young productive individual, approximately 1 in 150 individuals lost to the genetic condition, you can today treat them and they can continue being very productive members of the society. So I think that education gap is something that we need to cover very quickly.
Okay, sir. Thank you. The next question is, so there are many factors in the process of aging, like telomere shortening, damage caused by oxidants, etc. My question is, can we manipulate these things and live longer than an average human lifetime?
Yeah, very good question. So what a large-scale study in one of the Scandinavian countries have shown is that if you had a genetic diagnosis very early on, you can prolong your life today, by today's level of evidence, by approximately 1.5 years. I can share this paper in this group. Of course, you might not think that is very remarkable because 1.5 years of advancement in age of a population is not very significant on an average. But just think of it, that it took almost 70 years for a country like us to advance our age to what is said, almost 72, 73 today. And just one pandemic has demolished the advancements in age by almost a decade. Now, a few years of addition to an individual might not really become significant, but think of a few years of addition to a large population of individuals would mean a significant gain of productive life for the population. So I think on a population scale, this is possible to do today, and it is increasingly going to be possible as more and more genetic diseases become treatable or manageable.
Okay, sir, thank you. So, is there any questions?
It is already available in India, and even as we speak, it is available as a test. A lot of clinicians do not really understand the relevance of the test or importance of the test. But in fact, the test is available today. There are patients, and in fact, individuals who are not patients who are taking this test as it is. Absolutely. Absolutely.
One point, has all questions answered? All questions raised. I saw Archit Iyer and Adil Prajesh wanting to ask questions. Archit Iyer's question answered? Okay. Then fine. Okay.
Yeah, so to add on to that, what I can also say is a significant number of individuals in a population, approximately, if you put in that number, approximately 1 in 300 to 1 in 200 individuals in the population do have genetic variations which predispose you to developing cancer. This is called hereditary cancer syndrome. There are a variety of cancer syndromes now known, but each of them, for example, the most prevalent ones are BRCA1, BRCA2 for breast and ovarian cancer. Then there is colorectal cancer, which are very prevalent. Now, a significant number of individuals in the population do have predisposition to these disorders. That means that you can pretty much predict that the individual with a lifetime is going to develop cancer. For example, if you have a BRCA1, BRCA2 mutation, your lifetime risk of developing cancer, breast, ovarian, or for that matter, endometrial, or many other cancers, including prostate and thyroid, is extremely high. And this is approximately 87 to 90%. Now, if you know that you have that particular genetic variant, then of course you can modify your lifestyle to prevent the disease from occurring, and more importantly, institute early detection methods so that you can pick up these cancers very early on before they end up in advanced stage. So therefore, there is enough evidence to suggest that such early intervention, early screening, early detection modalities can extend the life of an individual, the productive life of an individual, to quite some extent.
Okay, sir. Thank you. I understood many about the increasing. I saw many videos about it increasing the lifetime of human life, but now I clearly understood what you have said right now. Thank you, sir.
Thank you.
Sir, am I audible? First of all, a big thanks from our part, sir. It was a wonderful session. My duty is to perform vote of thanks. So, I thank Vinod Scariya, sir, for his wonderful session on genomes. Then, the welcome speech is done by Navneet, is also a coordinator for our Space Club. Thank you, Navneet. And the Q&A session is done by Sharon. Thank you, Sharon. And I thank Alvin for the webinar quiz. Kuti sir, thank you for your valuable work. I thank Shalu sir for his valuable words also. Then Sandesh sir, Mithun sir, and Susan ma'am, thank you for your valuable presence for this session.
Hello, sir. Yes. Sorry, sir, I am at grade. Hello, sir. എനിക്കൊരു ക്വസ്റ്റ്യൻ ഉണ്ട്. അതായത് ഞാൻ ഇങ്ങനെ കേട്ടാണ് ഇൻമോർട്ടൽ ജെല്ലി ഫിഷ് ആണ് ജെല്ലി ഫിഷ് ഉണ്ട്, അതിന് മരിക്കലില്ല. അതായത് എന്ത് കാരണവശാലും മരിക്കൂല എന്നല്ല, അതിപ്പോ വയസ്സായി, ഐ മീൻ അത് ഓൾഡർ ആയിട്ട് പിന്നെ അത് മരിക്കും തോന്നിക്കഴിഞ്ഞാൽ അതിന്റെ പ്രായം കുറച്ച് കളയുന്ന എന്തോ ഒരു DNA പവർ ഉണ്ടായിരുന്നു, എന്തോ റീജനറേഷനോ. അപ്പൊ അതുപോലെ ഹ്യൂമണിനെ പറ്റാത്ത എന്തുകൊണ്ടോ?
ഹ്യൂമന് പറ്റില്ല എന്നില്ല. ഹ്യൂമന്റെ പല ഓർഗൻസും അതേപോലെ റീജനറേറ്റ് ചെയ്യുന്ന ഓർഗൻസ് ആണ്. നമ്മുടെ സ്കിന്നാണ് ഏറ്റവും വലിയ എക്സാമ്പിൾ. നമ്മുടെ സ്കിൻ ഓരോ ദിവസവും സെൽസ് മരിക്കുന്നുണ്ട്, അതേപോലെ തന്നെ നമ്മൾ റീജനറേറ്റ് ചെയ്യുന്നുണ്ട്. നമുക്ക് മനസ്സിൽ പോലും ആവുന്നില്ല നമ്മുടെ സ്കിൻ ചേഞ്ച് ചെയ്യുന്നുണ്ട്. നമ്മുടെ കോളറക്ടൽ, അതായത് കോളൻ അല്ലെങ്കിൽ ഗട്ടിലുള്ള എപ്പിത്തീലിയം അതേപോലെ റീജനറേറ്റ് ചെയ്യുന്നുണ്ട്. അതുകൊണ്ട് നമ്മുടെ കുറെ ഓർഗൻസ് ഇതേപോലെ റീജനറേറ്റ് ചെയ്യുന്നുണ്ട്. നമ്മൾ ലിവർ ഫോർ എക്സാമ്പിൾ, ലിവർ നമ്മൾ മുറിച്ച് മാറ്റി വെച്ചാൽ ചെറിയ കഷ്ണത്തിൽ നിന്ന് നമുക്ക് ഫുൾ ലിവർ റീജനറേറ്റ് ചെയ്യാൻ പറ്റും. പക്ഷെ നമ്മുടെ എല്ലാ ഓർഗൻസും അതുപോലെ റീജനറേറ്റ് ചെയ്യാൻ പറ്റുന്നില്ല, അതുകൊണ്ട് നമ്മൾ മരിച്ചു പോകും. അപ്പൊ കോംപ്ലിക്കേഷൻ കുറവുള്ള ലൈക് കോംപ്ലക്സിറ്റി കുറവുള്ള ഓർഗാനിസത്തിന് കുറെ അധികം ഓർഗൻസ് ഇതേപോലെ റീജനറേറ്റ് ചെയ്യാൻ പറ്റും, അതുകൊണ്ട് അവർക്ക് സർവൈവ് ചെയ്യാൻ പറ്റും. ഹ്യൂമൻസും ഇതേപോലെ റീജനറേറ്റ് ചെയ്യുന്നുണ്ട്, പക്ഷെ നമ്മുടെ ഒരു വളരെ കോംപ്ലക്സ് ആയിട്ടുള്ള ഒരു ഓർഗാനിസം ആയതുകൊണ്ട് നമ്മുടെ എല്ലാ ഓർഗൻസും ഒരേപോലെ റീജനറേറ്റ് ചെയ്യാൻ പറ്റുന്നില്ല, അതുകൊണ്ടാണ് നമ്മൾ മരിച്ചു പോകുന്നത്.
ഹലോ സാർ, എനിക്ക് ഒരു ക്വസ്റ്റ്യൻ കൂടി ഉണ്ടായിരുന്നു. അപ്പൊ സാർ പറഞ്ഞിരുന്നല്ലോ ചില ഭാഗത്തിനൊന്നും റീജനറേറ്റ് ചെയ്യാൻ പറ്റൂല. അപ്പൊ ഞാൻ ചോദിക്കുന്ന വെച്ചിട്ടുണ്ടെങ്കിൽ ഈ ഇമ്മോർട്ടൽ ജെല്ലി ഫിഷിന്റെ എന്തെങ്കിലും DNA പോലത്തെ എന്തെങ്കിലും ഒരു ഭാഗമോ അല്ലെങ്കിൽ വേറെ അക്സലോട്ടിലോ അങ്ങനെ സ്റ്റാർ ഫിഷ് പോലത്തെ ജീവികൾക്കൊക്കെ റീജനറേഷൻ പവറുകളുണ്ട്. അപ്പൊ അതുപോലത്തെ ജീവികളുടെ DNA നമ്മുടെ DNA ആയിട്ട് എങ്ങനെ കണക്ട് ചെയ്യാനോ അതോ അതിന്റെ പവർ നമ്മൾക്ക് ഉപയോഗിക്കാൻ പറ്റിയാലോ അങ്ങനെ എന്തെങ്കിലും ചെയ്യാൻ പറ്റുമോ?
അത് ചെയ്യാൻ പറ്റും. അതൊരു വളരെ നല്ല കാര്യമല്ല. കാരണം അങ്ങനെ റീജനറേറ്റ് ചെയ്യുന്ന ടിഷ്യൂസ് ആണ് ഇവഞ്ച്വലി ക്യാൻസർ പോലെയുള്ള രോഗങ്ങളാവുന്നത്. ക്യാൻസർ എന്ന് പറഞ്ഞാൽ ചില ടിഷ്യൂസ് നമ്മളുടെ എന്താ പറയാ മരിക്കാതെ റീജനറേറ്റ് ചെയ്തുകൊണ്ടേയിരിക്കുന്നു. അതുകൊണ്ട് പ്രത്യേകിച്ച് ഗുണമൊന്നുമില്ല. ബട്ട് ഇൻഫാക്ട് ഇറ്റ് ഇസ് അൺഅഡ്വൈസബിൾ ടു ഡു ദാറ്റ്. അപ്പൊ നമ്മുടെ എല്ലാ ഓർഗൻസും ഒരേപോലെ റീജനറേറ്റ് ചെയ്യണം എന്ന് പറയുന്നത് കൊണ്ടും കാര്യമില്ല. കാരണം ചില ഓർഗൻസ് നമ്മൾ റീജനറേറ്റ് ചെയ്താൽ ഇറ്റ് വിൽ എൻഡ് അപ്പ് ഇൻ ബിക്കമിങ് ക്യാൻസർ.
സാർ
മോനെ മെയിൽ അയച്ചോളൂ മോനെ. പ്രജീഷ് മോനൊരു മെയിൽ ഉണ്ടാക്കി അയച്ചോളൂ കേട്ടോ. ഓക്കെ
ഓക്കെ സാർ.
Thank you very much. Thank you very much.
Thank you so much.
Thank you.
I think we will have some occasion to meet and looking forward to have a glimpse of you, sir.
Sure, sir. Thank you very much, sir.
Thank you.
Okay, so nice.
It's a pleasure.
Thank you, sir.
Shajil Mash, I think